Today we present an article from Science entitled Ms4a7 expression in cDC1s determines cross-presentation in antitumor immunity. Type I conventional dendritic cells, cDC1s cross-presenting tumor. Antigens are essential for initiating CD8 plus T cell-mediated antitumor immunity. Although cDC1s possess this capability, their cross-presentation capacity is not intrinsic, but rather dependent on cues such as inflammatory signals or damage-associated molecular patterns. However, how tumor-derived signals regulate cDC1 function to shape CD8 plus T cell activation remains unclear. In this study, we identified Ms4a7 as a key molecule upregulated in cDC1s during tumor material uptake and nuclear factor B signaling activation, demonstrating that Ms4a7 is essential for effective cross-activation of CD8 plus T cells and antitumor immunity. The research team first inoculated mice with thymoma cells expressing GFP and ova, subsequently isolating and characterizing antigen-presenting cell APC subtypes. Diffless DCS exhibited cDC1 specific gene expression profiles. Differential genes were identified, followed by Cas9 screening and OTMINUS1 cell activation assays. Knockout of the Ms4a7 gene significantly suppressed cDC1-mediated CD8 plus T cell expansion, while having no significant effect on cDC2-mediated CD8 plus T cell expansion. Subsequent testing with diverse exogenous stimuli confirmed that Ms4a7 is expressed in cDC1s upon encountering tumor cells or exogenous stimuli. Subsequent RNA sequencing revealed markedly increased NF signaling activation in cDC1s. Upon FLA-cDC in LPS stimulation, inhibition of NF suppressed Ms4a7 upregulation, indicating that Ms4a's EVE-NS function in cDC1s is likely mediated via the NF pathway. Defects in Ms4a7 did not significantly affect antigen-presenting molecules or T-cell activation, and T-cell activation remained normal during direct peptide presentation. However, in experiments presenting OVA antigen, Ms4a7 divided by cDC1 exhibited impaired cross-presentation and was irrelevant to the cross-presentation process in mock knockout and overexpression studies. Further confirmed the specific role of Ms4a7 cDC cross-presentation. Subsequently, overloaded B2M divided by splenocytes were transferred into WT, Ms4a7 divided by, and BATF3 divided by mice. Assessment of OAT and OAT cell proliferation indicated a defect in cross-presentation. Ms4a7 divided by mice in vivo. Subsequent experiments in tumor model mice assessed T-cell activation in Ms4a7 deficient mice, ultimately confirming that Ms4a7 is essential for cDC1-mediated CD8 plus T-cell activation and in vivo antitumor immunity. Subcellular localization studies revealed that Ms4a7 predominantly resides in late endosome-divided by lysosome-associated vesicles, exhibiting high colocalization with RAB7, LAMP1, and RAB4-3, suggesting its involvement in vesicular transport associated with antigen processing and cross-presentation. Immunoprecipitation mass spectrometry identified interacting proteins, including CYP-NOX2, that are associated with transport and cross-presentation, supporting this mechanism within the tumor microenvironment. Ms4a7 deficient cDC1s exhibited reduced expression of pro-CD8 plus T-cell cytokines such as IL-12, IL-18, IL-27, impairing their ability to activate OTICD8 plus cells. Supplementation with these cytokines partially restored function. In vivo and in vitro migration assays demonstrated that Ms4a7 facilitates antigen-bearing cDC1 migration to draining lymph nodes, with Ms4a7 deficiency reducing the abundance of antigen-bearing cDC. one in specific CD8 plus T cells and DLNS. Collectively, Ms4a7 modulates antitumor CD8 plus T cell immunity by interacting with molecules involved in vesicular transport and cross-presentation, thereby influencing antigen presentation, cytokine production, and migration in cDC1. Single-cell sequencing revealed that Ms4a7 is predominantly enriched in the cDC1 population, with Ms4a7 positive cDC1 cells showing enrichment in pathways related to antigen processing and cross-presentation. However, expression was detected in some but not all subpopulations, reflecting functional heterogeneity of cDC1 cells due to the complexity of the tumor microenvironment. This was validated by immunohistochemistry MISI in bladder and colorectal cancer BLCA and CESC patient samples. Not all XCR1 plus CLEC9A plus cDC1 cells expressed Ms4a7, with Ms4a7 plus cells constituting a subset of this population. Approximately 33% of BLCA samples and 48% of CESC samples contain Ms4a7 plus cells. Further analysis of lymph node samples revealed that Ms4a7 plus cDC1s were significantly enriched in tumor-adjacent draining lymph nodes, particularly non-metastatic DLNS, whereas they were less abundant in distant or metastatic lymph nodes. Overall, high Ms4a7 expression is confined to a distinct cDC1 subpopulation, with these Ms4a7 plus cDC1 potentially performing more active antigen presentation and antitumor immune functions within tumors and their draining lymph nodes. That's all. Thanks for your listening.